Hello and good afternoon. My name is Neil Wary. I'm a senior at the Illinois Mathematics and Science Academy, and I'm very excited and honored to be presenting my research titled Connecting the Chromatin Remodeler CHG7 and the Regulation of Charge Syndrome and Autism. First, I'll go into some background on what charge syndrome is and talk about some possible connections among charge syndrome, autism, and cardiovascular disease. Then I'll go into the rationale and goal of my project. Following that, I will go into the key method I used, CRISPR-Cas9. Afterwards, I will go into the results and the implication of my results. To end, I will go over my summary and significance and my possible future plans. Now, a lot of you may be wondering, what is charge syndrome? Charge syndrome is a cluster of life-threatening genetic disorders, often affecting multiple organs. Children with charge syndrome may show the following phenotypes: coloboma, or a hole in the eye; heart defects, atresia of the choana, or blockage of the nasal passage; retardation of growth and development; genital hypoplasia; and ear anomalies or deafness. Most recently, it's been shown that a subset of charge syndrome patients are also developing autism. As I'd mentioned before, charge syndrome is a genetic disorder, and mutations in the chromodomain helicase DNA binding family of proteins causes the disease. The CHG family of proteins are chromatin remodelers. What that means is that this protein can bind onto DNA and histones and control the level of expression of several genes by twisting and turning the chromatin. Specifically pertaining to charge syndrome, it's been shown that mutations in the CHG7 gene, an ATP-dependent chromatin remodeler, is the main cause of the disease. The CHG7 gene is found on human chromosome number 8, and it's been shown that mutations in the first exon of this gene is the location of most of the mutations associated with the disease. For that reason, I chose to study the first exon of the CHG7 gene in my project. Charge syndrome is a neglected disease. Thus, we understand very little bit about the function of the CHG7 protein. Additionally, we do not know many of the genes that it might control. In my project, I proposed and hypothesized one possible function of the CHG7 protein. As I'd mentioned before, CHG7 is a chromatin remodeler. That means it can bind onto DNA and histones and control the level of expression of several different genes. One gene I took a look at is the vascular endothelial growth factor receptor 2, or VEGFR2. VEGFR2 can be found in endothelial cells, which are cells that line the blood vessels, cardiomyocytes, which are heart cells, and neuronal cells, or brain cells. The main function of VEGFR2 is to mediate angiogenesis in blood vessels, or the formation of angiogenesis in endothelial cells, or the formation of new blood vessels. Literature has shown that the development of the heart and the development of the brain are dependent on the formation of new blood vessels, so you can imagine the importance of VEGFR2 in endothelial cells. For that reason, I chose to study endothelial cells in my project. I proposed and hypothesized that loss of function of CHG7 protein in charge syndrome patients leads to decreased expression of VEGFR2 in endothelial cells. This thereby impairs the formation of new blood vessels in endothelial cells and eventually will impair the formation of the brain and the formation of the heart. My model could be a possible explanation for many of the phenotypes we see in charge syndrome patients, autistic patients, and possibly cardiovascular disease patients, as well as be a possible location for treatment of the diseases. To test my model, I wanted to mimic it in vitro, or in a petri dish. To do that, I used CRISPR-Cas9 technology. CRISPR-Cas9 is a novel genome editing tool, originally found in bacteria as a defense mechanism. The CRISPR-Cas9 system is made up of two main components: the Cas9 nuclease, which guides the gRNA to the DNA and cleaves at one specific sequence, and the programmable gRNA, which is complementary to the target DNA sequence of interest. After designing a gRNA, the gRNA will guide the Cas9 to the DNA, and the Cas9 will cleave the DNA at the specific site. After that, the cell's internal repair mechanisms can either insert or delete nucleotides, thereby inducing a mutation in the gene. In my project, I chose to create a gRNA that's complementary to the CHG7 gene. Thereafter, I could use gRNA and Cas9 to induce mutations in the CHG7 gene. After doing that, I will have cells that mimic the disease or have a disease in a dish model. As I'd mentioned before, I chose to study the first exon of the CHG7 gene in my project because that's where most of the mutations associated with the disease are. Here, you can see one of the three gRNAs that I designed, complementary to the exon 1 CHG7 gene. After editing, I wanted to confirm that CRISPR-Cas9 mediated editing had occurred, so I designed two oligonucleotide primers that would confirm CRISPR-Cas9 mediated editing to be later confirmed with PCR and DNA sequencing. After creating 3 gRNAs, I wanted to confirm that they were homologous to the CHU7 gene or complementary, so I used the BLAST tool to confirm that I'm editing the correct location. Afterwards, I needed a way to deliver the CRISPR-Cas9 system into endothelial cells. To do that, I used an all-in-one P lentivector. And after amplifying the plasmid with all-in-one P lentivector, I took the plasmid and transfected that into a packaging cell line. That packaging cell line then secreted a virus, and with that virus, I could infect endothelial cells, and the CRISPR-Cas9 system could start doing its work. Here is the first confirmation test that CRISPR-Cas9-mediated editing had occurred. The wild-type PCR product is around 792 base pairs in length. And after growing several clones, I saw that some clones had noticeable deletions. For example, a 292 base pair deletion. This was the first piece of evidence that CRISPR-Cas9-mediated editing had occurred. Afterwards, I wanted to confirm even more that this editing had occurred. And to do that, I did DNA sequencing. This is one example of my DNA sequencing. As you can tell, compared to wild type, one of the mutant clones had a 15 nucleotide deletion. For the purpose of my project, I focused on 2 clones. One of them had a haploinsufficient mutation, or one allele was mutated. The other had a biallelic mutation. After confirming using DNA sequencing, I wanted to see if the level of protein expression was also changed. Compared to control cells, you can see that the monoallelic mutation has a decreased expression of CHU7 protein. And in the biallelic clone, you can see a modest decrease in the expression, a severe decrease in the expression of CHU7. However, I wanted to see if after re-expressing the CHU7 DNA back into the biallelic clone, if I was able to re-express the level of protein expression of CHU7. And I was able to regain the expression of CHU7 after re-expressing that. Additionally, I wanted to look at the protein expression of VEGFR2, that protein involved in angiogenesis. And compared to control cells, you can see that both clones showed a modest decrease in expression of VEGFR2. However, after re-expressing the CHU7 DNA back into the biallelic clone, I was able to rescue the expression of VEGFR2, indicating that CHU7 is mediating the expression of VEGFR2. Additionally, I took a look at 2 apoptotic factors, P53 and P21, and showed that P53 and P21 show an increase of expression in CRISPR-Cas9-mediated editing of the CHU7 gene compared to control cells. Since P53 and P21 are cell cycle inhibitors, I wanted to take a look at apoptosis. And here is the apoptosis assay I performed. After plating and staining cells, I looked at the number of nuclear fragmentations to see if there is apoptosis occurring. And compared to control cells, you can see that the monoallelic clone had an increased number of nuclear fragmentations, and the biallelic clone had an even more increased number of nuclear fragmentations, indicating that patients with CHARD syndrome might be undergoing more programmed cellular death. Additionally, I performed a wound healing assay or a migration assay. As you can see, these first 2 lanes are my control, and the monoallelic deletion showed a decreased ability to wound the heel. On the right, you can see the quantification for this experiment. This was the first indication that perhaps angiogenesis might not be occurring in CHARD syndrome patients. This last and final experiment was my most important experiment. This experiment is called the branching point experiment, or the tube formation assay. It's an in vitro correlate of angiogenesis. And as you can see, in these control cells, and compared to the CRISPR-Cas9-mediated editing, there's less branching points indicated by each arrow. What this means is that patients with CHARD syndrome may be undergoing less angiogenesis. However, after re-expressing the CHU7 DNA back into the biallelic clone, I was able to rescue the function of angiogenesis in these cells. This means that CHU7 is mediating this very important function in these cells, angiogenesis. So what does this all mean? I've showed that a protein, CHU7, involved with CHARD syndrome is regulating a very important protein called VEGFR2, mediating angiogenesis in endothelial cells. Because the formation of the brain and the formation of the heart are dependent on the formation of the blood vessels, this could be very important if we want to understand autism and cardiovascular disease. In terms of future plans, I hope to do RNA sequencing to look at other differentially expressed genes. I'd like to perform chip sequencing to see if the CHU7 is controlling any other proteins. I'd like to study this in an animal model and possibly look for small molecule pharmacotherapies to treat CHARD syndrome and possibly cardiovascular disease and autism. These are my references. And I'd like to thank Dr. Kishore from the University of Illinois Chicago, the Illinois Mathematics and Science Academy, my family and my friends, my peers and my mentors for all their help, the Siemens Foundation, George Washington University, and Discovery Education for giving me this opportunity, and of course, you judges for listening to my presentation. Thank you.